to this meeting on that topic. I wanted to just thank people. So I did very little, which was the ideal way to organize this sort. Ben Domingue, who is a colleague of mine at Stanford and who is there, and David Rehkopf, who's over there, helped a lot in terms of actually finding people and inviting them. And Liz Vasile, who's there, did basically everything else, which is perfect. And these are the organizations that supported us financially. I couldn't resist showing you that we do have water in California. That's Shasta Lake, and it hasn't been this full in 6 years, so I'm glad. There's a thing. Okay, so let me, what I would like to do is just bring up some issues. Different people in this room are familiar with some of these issues, but perhaps not with all of them. And you may or may not share my point of view about some of these. But I think these are, or they represent, the constellation of things that I am intrigued by and challenged by as I think about the subject of this meeting. So, in general, the way I look at it, we observe the phenotype of an individual at some age and some time, and this is a function of the phenotypic history of the individual, the ontogeny, the developmental process. So it's a function of what happened to that particular phenotype in the past, which is where the minus signs come in. It's also a function of what happened to other phenotypes of that individual in the past. And then the genotype, the external environment that the individual lives in, and the internal environment of the individual. And this is a process that integrates over history in some way. The phenotypic part and the ontogeny, I think you are all familiar with. For the external environment, we've got hierarchies. So we've got the individual environment, we've got family, society. We may have larger forces at work. The genotype, we tend to think of genes in a lot of this discussion as if they were simply handed down from on high. And they are not. The genes are actually dynamic entities, and they change expression through the course of life. They change epigenetic state. And this is, I think, particularly important and is generally ignored in a lot of this discussion. They change their network state. That is, you are born with a complement of genes, and they don't do the same thing throughout life. Early in life, they are building an organism, and they are doing differentiation and things of this sort. And then, once that's done, then they have to sort of turn their attention to other matters. And as you get cellular differentiation, genes are doing different things in different parts of the body, and so on. So that's what I mean by network state. And then very often we are concerned not with it. Well, it's hard to distinguish between what we mean by a phenotype, but typically we measure phenotypes as things that we can grab an individual and measure their blood pressure or their weight or their height or something of that sort. And then we have some outcome of interest, for example, fertility, and we want to see what this relationship might be. It has become very common to use genome-wide association studies to do this kind of analysis. And I wanted to say just a little bit about that. In these studies, you actually look at what are called single nucleotide polymorphisms. So you're looking at a string of beads along the genome. You choose some number of them. You choose some number of people, and then you do this for some number of phenotypes. The number of SNPs that you choose is actually technology-driven. It's dependent on Illumina or somebody like that. And they, of course, work back and forth with the academic community At the present time, we have somewhere between 1 billion and a few million of these SNPs. The genome has well over 1 billion SNPs, so we're sampling maybe 1 in 10, 1 in 20, something like that. The number of people is cost-driven, and so we're getting maybe 10,000 if we're lucky. And if we do meta-analyses, we collect several of these things together. And the number of phenotypes depends a lot on a bunch of factors, and perhaps might go up to 1,000 or something like that if you put pooled studies together. There's a lot of attention to heritability, and I think it's overblown, but we will have opportunities to discuss this later on. The heritability is basically just an attempt to find out how much of the observed phenotypic variation is due to genes in some specific fashion. So it's actually estimated under fairly special conditions. It's not usually a useful or reliable guide to evolutionary change. Estimation is problematic. That's something we'll discuss. And there's no or little biology involved in the process of thinking about it. Here's a quote from Marilla, Sheldon, and Kirk, who are three of the people who do a lot of work with natural populations in the field. And this is a typical observation. You find high heritability, you find genetic variation, you find evidence for strong selection, and nothing actually happens. And this suggests that any number of possibilities. Either you're looking at the wrong thing, or there's countervailing selection that you don't know is going on, or there's selection on correlated things that you haven't actually probed, or something of the sort. But this is fairly widespread, and it's a puzzle. The one place where this actually does not happen is animal breeding programs. But in fact, as far as I know, in most of our humans' work, we are not interested in animal breeding or human breeding. That's neither here nor there. This is a quote from a very nice paper by a guy called Remington in Evolution last year. And it is a fact that I think might be relevant to the sort of things we do. Other factors, so in particular he points to the molecular architecture. And that's one of the directions that I've been thinking about a lot. Other factors that we need to think about are ontogeny, which is a matter of time scales, and I'll talk a bit more about that. The nature of selection, whether it happened a long time ago or recently, and environments. So you often see this statement made, and it's a reasonable enough statement. I mean, that's why we're doing this stuff. But in terms of the architecture of these things, where are these SNPs? How are these loci biologically relevant? And a lot of this stuff now can be done by actually taking a large number of phenotypes, finding out what the significant SNPs are, and finding out where they are on the chromosomes. You just line them up. Then you look at those chromosomal regions and you try and find genes that are close enough, defined according to some linkage metric. And then you say, what do these genes do? And does it make sense that those things should show up as in this list? And then the question of regulatory versus structural regions of the genome. So a lot of genes are, in fact, regulatory. And what they do is they regulate other genes that are the structural genes. So they act as catalysts, shunts, they damp throughput, and so forth and so on. And there's various ways in which you can do this. And one of my colleagues at Stanford, Hunter Fraser, has looked at a number of these kinds of studies in other organisms and some in people. And he argues that most of the SNPs that we find that are significant are in regulatory regions and not structural regions of the genome. That is an interesting phenomenon because you can then use these locations to formulate testable hypotheses about function, gene function, the function of collections of genes, gene networks. You can use, for example, convergent functional genomics. There's a nice paper out where you see similar networks in operation in C. elegans and in people and in flies, and you say, okay, that's a form of evidence that this hypothesis might actually be useful, and so we can do experimental tests of it in one organism, try and infer what happens in other organisms. We can make predictions about genetic correlations between phenotypes based on where these genes are, and environmental correlation. And again, the question of whether you're looking at regulatory genes or not tells you a lot about whether you expect environmental correlation. I think there's a lot of work to be done in terms of comparing countries, and I'm hoping that we can start doing some of this stuff. If you look at populations within countries, there's a lot of discussion, I think, not always very clear-cut, about the role of migration, how diverse populations are, whether it's due to old or new migration, whether it's due to heterogamy and things of this sort. And I think we actually have some evidence on a lot of these points from other studies in demography and so on that we can draw on. If you look at homogeneous versus heterogeneous populations, you can pick up signatures of important genes that you would not have otherwise. There's a very recent paper that comes out of Iceland and the Decode group on cholesterol, and they actually identified signals that they probably wouldn't have picked up in heterogeneous populations, and they're very excited about this because they want, of course, to make a drug and make lots of money. But I think these are interesting things that we can do. This is a subject that I'm really interested in, is the G by E interaction. And again, I think by doing comparative work, we can start probing this kind of thing. If we know, see, the trouble with a lot of our stuff is we don't have good measures of the environment. So we need to exploit and make the best use of proxies. And the proxies we can use are age. And we can start looking for not the stuff that's been done, which is you look at people 100 years old and 100 plus or something, and you say, let me find the 5 things that are in those in that population at high frequency, and let me then somehow deliver that kind of gene enrichment to people at age 40. That's not really the issue. It's more, if you look at people across the age spectrum from, all the way from 0 to 80 or something like that, are you seeing changes in SNP frequencies? Are you seeing, which you can relate somehow to cohort selection, to various specific forces that we've all thought a lot about. So that's what I'm talking about here in terms of enrichment. In terms of known environment, we actually spend a lot of time in social science talking about gradients. We've got gradients with respect to income, for example, that have been talked about a lot recently. And every time I see another study on this, the gradient widens. And it's at the point now where I'm starting to think that something must be wrong, because we are all supposedly getting poorer, and all the benefits are supposedly accruing to the rich, and yet everybody's living longer than they ever did before. So I'm not sure what's going on, but there is a very strong gradient effect, certainly. Where does that come from? Do genes have much to do with it? It's not clear to me that they do, but I think that's a natural experiment for us. And if we could take populations and divide them up into subsamples based on these kinds of known environmental variables, we might be able to say something about whether genes play a role and what role they play. David's going to talk more about G by E, and he's not going to talk about epigenetics today, I don't think, but that's another thing that people have done a lot of work on. There's the added health data, which Kathy's going to tell us about. And for me, one of the interests in it is this. Can you probe G by E interactions more effectively because you've got individuals that have been followed for a long period of time? And are there comparable data for other countries, and can you do something similar there? In terms of evolution, I wanted to just flash this picture up. This is a picture from my old boss, Dick Lewontin, and it's sort of a cartoon picture of how evolution actually works. You've got a genotype space up here, and these dots represent, maybe means for some population or something like that. And what you've got is the genotype, which is mapped into a phenotype. Here you've got the environment at work. Selection actually operates on phenotypes. It does not operate on genotypes. And it pushes the phenotype if there is selection in some direction. There are other processes that can affect this shift, but let's just talk about selection for a moment. These individuals now are the parents who are going to produce offspring, and so the gene frequencies that they deliver unto their offspring are different from those we started with. And then there are purely genetic processes here, recombination, copying errors and things of that sort, which shift the gene frequencies, and then the process carries on. And we are trying to infer what's going on in this space using movements in this space down here. One of the things that Lewontin was fond of saying was this. And if you think about it, it's actually a very important statement, because our picture of evolution has been that selection is weak and the gene frequencies don't change a lot, except when you have thousands of generations for them to change. And so, are we no different from the Romans? And if we really are no different from the Romans, then, all this stuff about modern environments and development and so on, changing gene frequencies, is just nonsense. I don't know whether this is true or not. And in order to try and determine whether this is true or not, we need to start thinking about the relationship between selection and evolution much more closely. So, neutral variation versus selective variation. How much variation in demographic traits is neutral versus selective? Don't really have much of a good idea. We can start thinking about the literature on selective sweeps and timescales. So this group, which is actually based at Stanford, has recently published a new method for looking at what they call singleton density scores, which is you find SNPs and you take a SNP of interest, and as you look away from that SNP along the chromosomes for a certain distance, and you count up how many singletons there are, how many non-polymorphic sites there are. That's the score. And this, then, they show, can be tied to this, whether selection occurred, whether there was a selective sweep in the recent past, and they're talking about, 100 generations or less, or whether it was a much longer time ago. And that's really important to us in terms of trying to understand whether genes are contributing in some way to the shape of what we see around us in terms of population structure. So I think there's a lot of stuff we could do, and we should start thinking about. Finally, since I'm fundamentally a modeling guy, there's a lot of questions related to these issues. which I think have not received a lot of attention, in my view, in the modeling literature. One of the things about population genetics that made it so significant as a field was it had a bedrock of well-understood models. And those models were developed over the years, and they became more sophisticated, and they gave us things that we could test our observations against. And I think in the newer development, we have a torrent of data coming down upon us, and we don't do much of this. It's very hard to get people to support any kind of actual thought about these issues, but I think it's worth doing. We don't have good G by E models, for example. We have a statistical model is not a model. It's just a formulation of a potential relationship, and you can get an estimate or not. Under certain assumptions about how the world works, it gives you certain conclusions. A theoretical model is very different. It actually makes a different set of assumptions about how the world works at much lower levels, and then tries to assemble a picture of what you are going to see at the upper end. And I think we don't have, for example, good G by E models. If you look at the entire evolutionary biology literature, there are actually only 3 out there, and none of them seems particularly useful for the kinds of contexts we study. Relatedness, I think we're going to have a talk which addresses some of this, but I don't really understand relatedness in the context of the software that's being used. Everybody uses Plink, and there's Beagle, and there's something else and something else. And if you read some of this, in one of these, there was a discussion of identity by descent. And the paper said something like this. We don't want really old identity by descent. We don't want sort of intermediate frame identity by descent. We want recent identity by descent. Okay, how are we supposed to tell them apart? They say, our software does this. I've tried to read through some of this software. It's really hard. Nobody seems to feel the need to write down the equations that generate the coefficients of relationship that are being produced by this software. I don't know where it comes from, and I suspect a lot of other people don't either. Identity by descent has been used for a very long time in genetics, and we use it at all sorts of time scales. We try to understand the tree of life with it. We try to understand the mammalian section of the tree, the primate section of the tree, the human section of the tree. And in all of these, we use that concept, identity by descent. I mean, we're supposed to share 99.9 or 99% or 97% of our genome with chimps. And it's not obvious to me that if we threw a few chimps into our GWAS study, we would pick it up. I mean, I asked, and I was told, yeah, sure, I'm sure it's going to happen. And I certainly hope so. It would be nice, but I don't understand. What is it? What signal is it in that software that is picking out these things? It's not clear to me. I'm entirely willing to believe that, as usual, I am simply ignorant, but I'm happy to learn. And so these are questions that I think we would do well to have somewhere in our list of things to worry about occasionally. And that's a long enough list of things to worry about. And I will end with this, which represents my picture of how science works.
All right, well, thanks for having me. I'm Dan. I'm here from Duke. Apologies to those of you who have seen this one before. There are a growing number of you in the audience. So this may be the last time I do this talk. I hope it's getting better each time. I'll give you just a very quick introduction to me, since Tulja very helpfully covered most of the introduction to genetics that makes up the front end of my slideshow. I study two things. I study how human life courses are assembled, and that's the work that I'll show you today. But I also study how human bodies fall apart. And so I guess that's a little plug to also ask me about aging. All right. Okay, so I'm going to do the acknowledgement at the beginning so I don't run out of time. I'd like to thank my collaborators at Duke and the Institute of Psychiatry, but also in particular the folks at the University of Otago in Dunedin, New Zealand, who collect all the data that I'm going to share with you today. And I'll also acknowledge Ben Domingue, who's in the audience today, who's part of this project. Okay, so this is the basic outline of the talk that you'll get. There's a lot there, so I'll try to move efficiently. Okay, so the background. I think we all study genetics for different reasons. To some extent, I study genetics because the questions I'm interested in, the phenomena that I'm interested in, are heritable. They're influenced by genetic differences between individuals. The DNA that we can study is somewhat special in the world of human science because it does come before almost everything else humans experience, although obviously if you go back generations, there are opportunities for other kinds of relationships. And then ultimately, the idea is that by learning something about how the DNA sequence regulates or affects human behavior and achievement, we can learn something about causal processes that we might intervene on. And I'll get into that a little bit more as we move forward. This group, I think, probably doesn't need this slide, but just very quickly, in human genetics, for a long time, the way the work was done was hypothesis-driven, where we would reverse engineer the biology of a phenotype to arrive at a protein. We would try and find a gene that encoded that protein. We would look for variations in that gene, and then we would try to relate those variations to back to the phenotype. These days, we do something a little different. We take a comprehensive survey, more or less comprehensive depending on the technology, of the genome, and we do hypothesis-free search. And that's the work that I'll share with you today is built on that hypothesis-free approach to understanding the genome. We all, I think, know what a SNP is, but in case you always wanted to know but were afraid to ask, that's what a SNP is. It's a single letter change in the alphabetic sequence of human DNA, and at some point in time a mutation arises, it proliferates, and now we have a 1 in 100 or more frequency of that particular variant, and that's what we're studying. Doing these hypothesis-free surveys of these single nucleotide variants struck some people as a preposterous activity for quite a while, but over the last decade or two, we've amassed a very large number of, what are equivalent to p less than 0.05 relationships between common variations in the genome, and basically any phenotype you can think of in biomedicine, and increasingly now into the social and behavioral sciences. So we're going to learn something, I think. I'm a glass half full guy, at least as far as this is concerned. The way we do GWAS, though, is a very brute force approach, right? We're going to conduct millions of hypothesis tests, and so when we correct for that large number of tests to get back to that 1 in 20 false positive rate, we have a very stringent p-value threshold that we have to meet. And the most effective way of meeting that threshold, it's turned out, is to amass extraordinarily large sample sizes. So we know a great deal of detail about their genome. We've measured a couple of million of these SNPs, and we know some kind of a phenotype, but often we know very little else about the individuals participating in these studies, which leaves lots of unanswered questions about the kinds of relationships that we observe. So then we have to do something once we've made these GWAS discoveries. And I think the conventional path to translation of a GWAS discovery in biomedicine is to try and build the chain from the bottom up. So we start with this sequence variant. We're going to trace that to some difference that occurs in transcription onto protein production and on upward to phenotype. The idea there mostly is we're going to find a drug target. We're going to hit something with a pill and we're going to sell it for a lot of money. So that's not the way that I think about this. I'm doing this work to go backwards. What I call top down, right? But the idea is to trace a path backward in development from the observed phenotype in the GWAS to the process that has produced it. And the idea, again, is along the way to identify some process that we could manipulate to change the phenotypic outcome, although the difference is I'm not looking for a drug target. And that frees me up a little bit in how I can study the genome. So non-drug interventions are primarily what I'm interested in. We know that complex traits that are the subject of this meeting are not monogenic, right, by definition. But there's no gene for a particular trait or behavior of interest in the worlds that we work in. There are lots of them. And so there needs to be some way of wrestling that large number of variants, each making very tiny contributions to the ground. The way we do that these days, for the most part, is through polygenic scoring. And so this is a fairly simple method that takes the coefficients estimated from GWAS, weights the phenotype-associated allele of each SNP by those coefficients, and then sums them up. So you get a nice normal distribution that quantifies in some respect a level of genetic load toward a particular trait or outcome. And what I do then, and what I'd like to convince you is an interesting thing to do, is to take those measures of the genome that we get from these hypothesis-free surveys and examine how their effects play out across the course of human development using prospective longitudinal studies, preferably of populations that represent something of interest to us. So not samples of convenience from clinic populations, but populations that we think reflect the social problems that we might want to study. And through that process, we can learn something about how genetic effects operate, which in turn may be able to guide us toward some inference about cause and ultimately toward the refinement of intervention. So the work, I guess, that I've done in the past has been about biomedical phenotypes, asthma, obesity, smoking behavior, which is kind of on the border. But in 2013, very helpfully, the Social Science Genetic Association Consortium started to do GWAS of social science traits. And many of you in the audience are also sort of pursuing that project. And we have this GWAS of educational attainment, and now, in fact, we have EA2. The results I'll show you today come from EA1. The little bit of looking I've done at EA2, the results are the same. The effect size is maybe a little bit bigger. But before I get into it, let me put up this kind of a trigger warning. This actually was an article that was written before the EA2 came out. And I think it's important to recognize that when we begin to dig into the genetics of human achievement, there are some demons, there are some nightmares in the closet that we have to at least acknowledge are there, and we have to think about how we present that information because it exists in the context of this kind of an issue. I also thought I would put up this quote from Ed Yong about the new educational attainment. You can almost hear the tsunami of misinterpreted takes cresting on the horizon. The social science genetic, I'm going to call them the SSGAG because that's just a simpler way to do it. They've done a great job of emphasizing that small effects, tiny fractions of variance, not a path to testing who we should intervene on, who we should promote, or which fetuses deserve to be developed. So with that as context, I'm about to sort of grab something radioactive here and wave it around in front of you. I'm going to go ahead and do that. So I guess the first thing I'll observe about genetic associations with educational attainment is they're non-spurious. The findings of the educational attainment polygenic score have been replicated on a couple of continents, and in particular, and perhaps most importantly, in sibling comparison designs. And the reason this is important is that both genes and social position are passed along across generations. And so it could be the case that when you do GWAS of educational attainment, what you detect is a genetic signature of a privileged class that likes to breed within itself. When you study siblings who share identical ancestries, you can be assured that that is not the artifact that you're detecting, right? So, all right. So what does it mean to do top-down research then on the genetics of educational attainment? I think the first question I wanted to ask was, in fact, is that the top? Is success in schooling the end result? Because at the end of the day, we're interested in education not because we think everybody should be well educated, but because with education come all the good things under the sun in the modern world. So we wanted to begin with, do the genetics of educational success predict success beyond schooling? If they do, how does that process come about? What are the steps along the line that connect a child's DNA sequence at birth with its socioeconomic attainment decades later? And then finally, why does it work like that? And the why here, the mechanistic why, is the why from psychology. So what are the characteristics of these children that lead them to live these different lives which result in these differences in attainment? So now to the empirical part of the talk, this small island in the Pacific Ocean, where these 1,000 babies have been followed now for 40 years. We're going back to the field next year when they turn 45. It's a small cohort. It's special because it's all the babies born in a given hospital, the Queen Mary Hospital in Dunedin, New Zealand, 1972 to 1973. So it's representative, and it's intact. Across these 4 decades of follow-up, we have 95% retention of the surviving study members as of age 38. And in fact, this 95% isn't just that 96% minus 1. There's a couple of people who weren't there at age 32, came back at 38, and that's more or less how it's played out across follow-up. So essentially, we're observing nearly everyone at nearly every assessment. And that means that we can study the process of attainment without concern that we're observing a sample that has been restricted to only those who are a little bit successful or may miss the most successful. Everybody's in. To give you a sense of what 4 decades of follow-up look like, these are pictures of 2 of the study members taken at each of the assessments. So there they are at 38, and that, if you've ever wanted to know what a GWAS chip looks like, that's what it is. Okay, so this is the basic design. We're going to follow these kids from childhood through midlife, and we're going to investigate where they end up at the end, and then we're going to go back to the beginning and see how they begin to diverge from one another in terms of early developmental milestones, what they did in school, and how they behaved as adults. And finally, what it is that may drive that variation in life history. Okay, so this is the score. It's normally distributed just like you'd expect. It's all the SNPs that match between the imputed Dunedin GWAS and the SSGAC results list, so it's a couple of million. The analysis sample is slightly smaller than the Dunedin cohort. The indigenous New Zealand population, the Maori, don't allow their blood to leave New Zealand, and so they were not included in the genotyping. But it also means we have an extremely ethnically homogeneous cohort. We nevertheless, I'll report results that are adjusted for the first 10 principal components. You raised the issue of what are we adjusting for. I think Dalton's going to talk more about it tomorrow. The results are exactly the same if you don't adjust for these 10 principal components. In fact, if you're interested, the paper, we report results both ways. Okay, so here's the replication phenotype. Educational attainment in New Zealand for this birth cohort is a little different than what we might be familiar with in the United States. People had the option of leaving high school at age 15 with a leaving certificate, and this was a perfectly adequate credential to apply to college, to get a job. It was not like a GED, which is sort of not as good as a high school diploma in some sense. They could stay on and graduate from high school, and then they could go on and earn a university degree. So that's how we've divvied up the cohort here. And you can see that the plurality do the complete high school, but a good number of them, about 1 in 6, drop out of high school with no credential. About 1 in 6 take this leaving certificate, and then a little less than 1 in 3 ultimately earn a university degree. And these are the average polygenic scores for those 4 groups of kids. What you can see is there's a little less than a half a standard deviation swing between the dropouts and the baccalaureate credential. Yes?
I'm sorry, I just missed, you said before, about how many SNPs are included in this score?
A couple of million.
A couple of million actually.
That's right. So we used all the SNPs reported by the Social Science Genetic Association Consortium. We used no thresholding to exclude SNPs based on statistical significance, and that's based on evidence that score performance in terms of predicting phenotypic variation tends to increase as you relax the p-value threshold up to some asymptote, at which point score performance flatlines, but it doesn't decline. And the reason here is because we're weighting the contribution of each SNP in the score by its estimated GWAS coefficient. So the better the GWAS is, the better powered, the better job it will do at sorting the causal variance into the low ranges of the p-value distribution, and the better job it'll do at estimating the beta weights, the coefficients for those SNPs. So even in a GWAS that's done perfectly, in which all the SNPs that are causal are 10 to the negative 8th, okay, that are genome-wide significant, that same GWAS will estimate very, very small coefficients
essentially zero coefficients for all the other SNPs, and that's why leaving them in doesn't cost you anything in terms of score performance. But I think we're going to hear a talk later on about sort of the mechanics of polygenic scoring as well, so we can get into that then. Okay. So what is the outcome? How do we measure how far a person has gotten in life? We could measure what kind of job they have, and that's what an occupational prestige scale does. This is more or less the average income and education for a job assigned to a census coding of that job category and then linked to a person based on what they tell you they do for a living. There's how much money they make. The exchange rate between the United States and New Zealand has varied between about 1 and about, US dollar is 2 New Zealand dollars. So that 150,000 cap on personal income is not, doesn't give us a much capture of the high range of the income distribution. But we also have information about their assets that come from a structured interview about different things they own and how much those things are worth. We ask them about problems they may have had borrowing money, about problems. We actually have their credit scores from the Veda Credit Bureau, which is the largest credit bureau in New Zealand. We have their social welfare benefit use. So the number of days, and this is actually cumulative since they were, I think, 21, that they've used social welfare benefits in New Zealand. It's a very generous social welfare state. You can see, 2/3 of the cohort have used at least 1 day of benefits by the time they were 38 years old, difficulties paying expenses. So I'll show you each of these individually, but what we did first was roll them up into a factor score. So all of these things, as you can imagine, are correlated. They're correlated in precisely the way you'd expect. It doesn't come out so well here, but all the bad things are negatively correlated with all the good things. And in fact, the factor loadings for all these different items are about the same on this general attainment factor. So does a polygenic score trained on the educational attainment of 100,000 people predict how far these 1,000 people have gotten? Yeah, about as well as it predicts their education. So again, to echo the SSGAG, the effects are small. They're all small effects. But they are in the expected direction. So we see that kids with higher scores end up at age 38 with a higher level of socioeconomic attainment. Was that just because they have more schooling? About half the story is being explained by differences in the educational attainment of these individuals. I don't know how representative this result is because of the characteristics of this particular New Zealand birth cohort. Lots of them did not get college degrees and still got good jobs. Is that going to be the case today? Probably less so. Although there'll be much more heterogeneity and achievement among the college degree holders. What we see here is about half of the genetic effect that we measure for socioeconomic attainment broadly is explained by differences in education. And this is how it breaks down if you look at the unadjusted and then adjusted for educational attainment. These are effect sizes in terms of Pearson's R with the different items in that attainment factor. So you can see some of these associations are more explained by education, some less so. So this is all very interesting, but this is, I think, really the question that motivated me is how does this score relate to the social mobility of these study members? If you see a score that predicts educational attainment, it predicts socioeconomic attainment, we presume that the score is roughly the average of the two children's parents. We might expect that the ones that we see doing well were simply born into well-off families. So how much of this association is independent of the social origins of the kids? And in fact, I don't think I have this on a slide, but the correlation between the polygenic score and the occupational prestige of the study members' parents measured when they were kids is about the same as the effect size for the correlation with the study member's own attainment. So in the neighborhood of 0.1. All right, that's helpful. Right, so this is just a intergenerational transmission of social advantage slide where you can see that rich kids tend to grow up to be rich adults and poor kids tend to stay poor. This is a slide where you've got the original Blau and Duncan paper there, and then a GCTA and SNP-based analysis of this kind of intergenerational transmission of social position by Robert Plomin and his graduate student, maybe not graduate student anymore. And what you see here is that there's heritability to social position, and a portion of the association between your childhood social position and your performance in education is attributable to genetic factors. That's the claim. So this is what our data look like. So the adjusted association between the polygenic score and that attainment factor, if we count for childhood social position as a continuous variable, is more or less the same as it is unadjusted. So 0.13, 0.11, not much change. What I'm showing you in the graph here, just for visualization purposes, is that association stratified. So I'm showing you kids who were born into poor families, middle-class families, and wealthier families. And I've got about 500 and change in the middle and about 200 on either side. And just to orient you, first, you see the three clusters of dots are at different levels in each of the three graphs, right? And that's intergenerational stability, right? If you are born into a low-income family, you'll have a range of different outcomes, but on average, those outcomes will be poorer than kids who are born into well-off families. Okay, so that's what that dashed line in the middle is illustrating. All right. The second thing I'll show you at the bottom of the graph, these box plots are showing you the distribution of the polygenic score in each of these groups. So that's the gene-environment correlation that I mentioned earlier. You can see that kids born into wealthy families have, on average, above average polygenic scores. Kids who are born into poorer families have below average polygenic scores. So now the sort of brighter side of the story, the regression lines in the plots are showing you that no matter what station you are born into in life, having more education-associated alleles is associated with upward mobility. So you end up better off. These effects are independent of your social position. And then I guess a final point of orientation, these are bin scatter plots. So each dot is the average x and y coordinates for a bin of 10 study members. So the regression lines and the effect size estimated from the raw data, they're basically slightly more informative confidence intervals, I think is the way to think about it.
Sorry, what are the slopes of those lines? Do you have them by any chance?
I do have them. I don't have them off the top of my head. Yeah. Are they different is the question, right? Doesn't this one look a little steeper than that one? Statistically, it is not steeper. We didn't test that in the paper, but Dan Benjamin asked me to send him some results, so I did. So I know that it's not statistically significant. Dan estimates that about 10,000 people are needed to have reasonable power to test a G by E that has an effect like this.
I'm sorry, are those slopes for the original data or for these bin data?
Original data.
It does also appear that there's substantially more variation as you go up in the SES scale, or is that again just in terms of just the correlation between the points and the
right. I think if you were to see the raw data, you wouldn't.
Be so.
It wouldn't be as dramatic.
Ours are what 0.3, 0.4 in there, because in the low SES it looks more like 0.7 or 0.8.
But that's because of the binning, right? And because of the difference in the scale of the y-axis and the x-axis.
Okay.
These correlations are about 0.1.
0.1.
Okay. So given that we see this process, this social mobility, something we would absolutely like to engineer in the general population, how does it come about? So the first thing we looked at were a set of early developmental milestones that your pediatrician might ask you about when you take your toddler to the doctor. Are they smiling yet? Are they sitting up? Are they feeding themselves? Are they talking? All right, and these are survival plots. This is Kaplan-Meier. These are months, and this is the proportion of the cohort that hasn't yet reached that milestone that's being graphed. Okay. And this is now that same plot, and just for graphical purposes, I'm showing you kids with a standard deviation above the mean on the polygenic score in blue, and kids with a standard deviation below the mean in the polygenic score in red. If I graph the rest of them, you'd see a black line right in the middle of the red and blue lines. In general, what you see is that there are no differences in the timing to reaching these developmental milestones, with two exceptions. They talk a little earlier. It's a small effect, a little earlier, and they begin using sentence perhaps a bit earlier, which likely has to do with the earlier onset of talking. Fast forward a few years, we can look at how they learn to read. So the same reading test was given to these kids at 7, 9, 11, 13, 15, and 18 years. You can see they hit ceiling on it. Some of them obviously never score very well. And we can, again, show that stratified analysis. Do the kids with higher scores learn to read faster? They do. They're reading better already by age 7, and their reading performance improves more rapidly from age 7 forward, and they hit maximum earlier. Okay, so I've cut out the schooling for time purposes, but what I can tell you is that these kids, while they're in high school, select into standardized testing for more competitive thresholds. So more of them take, as an example, a bursary exam, which you do in the equivalent of your senior year of high school, in which you compete for scholarship funding. So kids with higher scores more likely to take this bursary test, and even among the kids who select into that bursary testing, kids with higher scores perform better on that test. So as we observe them moving through high school, we see them aspiring to higher levels of achievement and then outcompeting peers on the metrics that we observe. But I'm going to take you on the other side of high school now. What do they do afterwards? So in New Zealand, it's very common to take at least a year and go abroad. It's a small island in the middle of the Pacific Ocean. Get out, see the bigger world, swim in a bigger pond. What I'm showing you here are 1,000 very thin lines in segments equivalent to 1 year in length, all across the follow-up from age 18 to age 38. And a blue segment is a year spent outside of New Zealand. So what you can see is that we have some folks who've moved mostly to Australia. But that cluster of light blue lines in the middle is showing something else. It's called the overseas experience. And it's a classical marker of being on the fast track to success in New Zealand. So there's a nice Wikipedia entry on this for those of you who are interested. But the OE is how young New Zealanders distinguish themselves as headed for success. And these are the fractions of kids with polygenic scores -1 and below, within a standard deviation of the mean, standard deviation more above, who have an OE. Okay, and this is it's about a 17% increase in the likelihood that you're going to have an OE for each standard deviation increase in your polygenic score. So they're more likely to seek out this overseas experience, and they're more likely to stay gone. So brain drain is of great interest in the New Zealand government. This analysis has actually been performed before with intelligence test data from New Zealand, but here we were looking at whether the kids who migrated out, which we define as residing outside of New Zealand in a single place for a continuous year at the time of the age 38 assessment. And these are the differences between the mean scores for the groups in these different places relative to those who stayed in New Zealand. So kids with higher polygenic scores are migrating out. And again, about a 20% increase in the likelihood of being one of these migrants for each standard deviation increase in their polygenic score. The means here are sort of provocative, but I wouldn't interpret them too much. These are very small groups of people. We've got about 200 in Australia, but we're talking dozens in the other places. Okay, how were they at husbanding their resources? We measured their financial planfulness by asking them about whether they had investments, whether they owned a home, do they have a retirement account. They're a little bit more planful. We asked their friends about their financial problems, so the survey was given to a number of informants for each study member. They rated them as having more or less financial problems. If you have a higher polygenic score, your friends think you're better at managing your money. So there's some evidence that they're sort of engaging in some more planfulness there. Here we looked at their partners. So we know not a whole lot about their partners. For those who are in a committed relationship lasting at least a year at the time of this measurement, we get their income and their education. In this very simple SES scale, you get a point for having an income above the sex-specific median for New Zealand, and you get a point for having a university degree. And what you can see is kids with higher polygenic scores are selecting higher SES partners, or being selected by higher SES partners. Okay, so they're leading very different lives. What traits or characteristics might explain these different lives that these children are leading? So the social side, the SSGAT got into doing GWAS of educational attainment because they were interested in the genetics of intelligence, but you couldn't get enough samples with well-measured IQ. We've given the Wechsler, not me, I wasn't there, but the Wechsler IQ tests were given to these study members when they were 7, 9, 11, and 13. And so we have a very good estimate of their IQ. Here it's just on a standardized scale, but mean 100, standard deviation 15 is the traditional IQ metric. And what you can see is that kids with higher polygenic scores score higher on these IQ tests as children. And this effect is a little bigger if you look at EA2. What about non-cognitive skills that we hear a lot about as being important? So this is an index of their self-control. The measurement is a little complicated. There's a paper I can share with you about that. But this is the ability to delay gratification, to act planfully. And we see these kids have more self-control. And then finally, we wanted to know if they were sort of friendlier, nicer. And we don't have a great measurement of this, but what we do have are ratings of the kids made by the research worker at the study who walked them around to their different assessments when they were kids. So at every age, between 3 and 9, a research worker chaperoned the child around, and at the end of the day, made ratings of the kid. Were they friendly? Were they confident? Were they cooperative? Were they communicative? All right? Was this kid fun to be with? All right? And so we rolled that information up into a scale, and what we can see is that people liked the kids with the high scores more. So it's not just sort of being more clever. You also need to have a bit more self-control and be more likable to achieve the benefits that we're seeing. So on the whole, a majority of the genetic association with education was accounted for by those 3 characteristics, and slightly less in the overall socioeconomic attainment. Is that measurement error? Is that because there are other untapped factors? I would guess the answer is both. I'm running out of time here, though, so let me just hit this final point. One of the questions we had coming to these data was, all right, so these kids do better. They look better on a lot of important metrics, but we thought about this primarily as kind of a neck-up phenomena. We're looking at psychology when we measure intelligence, self-control, interpersonal skill. What if they're just kind of better built overall? Are they physically healthier as children? So we have a childhood health index that I put together from lung function, anthropometry, blood pressure, motor and balance testing that were conducted at each of the childhood assessment waves. And this is a pretty good predictor of adult health, as those of you who studied connections between childhood and adult health will know. There's no association between this polygenic score and the physical health of these children. Another thing that we did, someone suggested, well, look at the polygenic score for height and see if that predicts all these good outcomes, because we've seen the Case and Paxson papers that being taller gets you everything good in life that having more years of schooling gets you. The education polygenic score is not associated with the height polygenic score, as those of you who have the data will know. And the height polygenic score also does not predict differences in the attainments of our study members. Okay. So there are lots of limitations. These are a few of them. This, I think, is important. So GWAS of educational attainment are not just genetic discoveries for education. The psychology that underpins those genetic associations is more complex than simply raw candle power. And I think that's important in terms of thinking about what we want to cultivate in kids. I think that in the sort of weak associations we see with time to talking and reading, there is the suggestion that we may be able to move children's outcomes by intervening on these developmental precursors to successful lives, although I think we need different data for a better test of that hypothesis. To me, the most provocative thing about this is that the GWAS of educational attainment got panned by a lot of geneticists in the beginning for having a lousy phenotype. It was like, oh, what could be genetic about what's such an obviously social outcome? And to me, in the data that I've just shown you, associations with how people manage their resources, with how they breed and how they migrate, we're seeing something like a life history strategy difference between the lower scoring and the higher scoring kids. Now, is that true? I have no idea. But this is the first set of GWAS results I've looked at that has suggested to me something about the evolutionary biology of human individual differences. Okay, so I'll leave you with this one. Because I'm over time. But thank you very much for your patience.
Great, thank you. So I was happy to see that Dan and Melinda changed the name of their title. But I admit this was the original title, and then when I put the talk together, this is really what I'm talking about. It's pretty much the same thing. So we're going to switch gears a little bit here. I'm going to talk about the past, present, and future for data and the research that comes out of it on genes and human phenotypes from the lens of ad health. This is the topic, so not so much presenting any original research or methodology, work with a lot of people here, but more a sort of big picture step back from a data producer. And I also wanted to say, as a sort of caveat, that some of the comments that I have here might come across as critical, and to the type of analysis that, like Dan and Melinda just presented, which is the kind of research that we're hoping is going to come out of the type of data that we produce. So just keep that in mind. We have over 30,000 users of ad health data, and so that's where I'm coming from, as well as reading the literature. So I'm going to talk a little bit about sort of the evolution of genetic data to give you some background in ad health and then the research that it produced, so you can see sort of where we're coming from, a little bit of historical perspective. Then the challenges that come up, so you can think about, we're all familiar with different types of data, and we know the problems and the issues that come up with the data that we use. I'm going to talk a little bit about the genetic data. Now, what do social behavioral scientists care about, what do geneticists care about, and some of the limitations and recommendations. So if I talk about ad health, I need to give you a little bit of background on the design. So I'm going to zip through this quickly. But the unique aspect of ad health is the scientific objective was to try to understand the causes of health and health behavior among adolescents with a focus on the role of social context. So most of the research on adolescents, this was developed in the early 1990s, just focused on the individual and devoid of sort of the environments in which they live their lives. So that's what ad health, that's where they stepped in to play a role. So ad health was a school-based design. What we did is we selected schools. And when we select schools, then that means that we are selecting individuals who know each other in those environments. And we exploited that in terms of the design. So we had a sort of school administration, and then from the school rosters, we sampled adolescents at a rate of about 1 to 9. And then this adolescent cohort we interviewed in the home, and that represents the prospective cohort that we follow over time. So by sampling schools, that meant that we were able to characterize the school environment. We also interviewed the peers and friends and romantic partners of the adolescents in our sample, so we had information on those contexts. We also interviewed a parent, so that fills in the sort of family context. And then we merged in an array of geographic and spatial and built environment data. The reason I wanted to show this is, as part of this original design, we selected a main sample of 200 adolescents from each school pair, and then we have these oversamples. So we have some ethnic oversamples, a disabled sample, saturated samples, which means we just interviewed everybody in the home that were in a set of schools. And this gives us this sort of sexual network data. But as part of the original design, we have these genetic samples, and these are 300 pairs of adolescents who have these different genetic resemblance. And then this is the longitudinal perspective of ad health. So we've got 5 waves of data. We actually go into the field. I just got a upsetting email on June 6th. So is that 2 weeks? Yeah. With wave 5, we've been doing some pilot testing. But wave 1 occurred in 1994, 95, and these are the adolescents then who are in grades 7 through 12. And we had a little over 20,000 in this prospective cohort that we followed over time. Wave 2 was 1 year later. Wave 3, 5 to 6 years later. Wave 4, 5 to 6 years later, and now we're in the field again with wave 5. And then we have this other information. We interviewed about 85% of the parents at wave 1. We're currently doing a follow-up of about 500 of those parents. We have a couple sample where we interviewed a partner of the AdHealth respondent. These were quota samples, so 500 married, 500 cohabiting, and 500 dating. And we have these IIV studies are just our repeat measures of the biomarkers that we collected, including the genetic data that allow us to test for reliability. We're doing pretty well in terms of response rate, not as well as the median. That's really quite enviable. Trying to figure that out. But the last wave, they're now adults. They're 24 to 32. We found 80%. We always go back after the wave 1 respondents. They have a credit record now. We can find them a little better than when they were in this transition adulthood period. And at wave 5, they're now going to be moving through. Oops, I forgot to update that. Their 30s. Okay, so the data. So we began then with this embedded sample of 3,000 pairs of adolescents with some biological resemblance. So from the outset, the design of AdHealth was to include this important aspect of biology and trying to understand health. But our main focus at that time really was on the environmental effects. And of course, we knew that I joined AdHealth as an assistant professor and wrote the grant. I'm a family demographer, and I wrote the grant that was interested in family effects and parenting. And so we know that our family effects have been overstated, due to shared genetic variance between parents and children. And so our interest really was trying to control, get rid of that so that we could really get a better estimate of the environment. At the time, we were thinking mainly about just basic behavioral genetics models. One of the co-investigators on AdHealth was David Rowe, who was a behavioral geneticist at University of Arizona. The methodology, of course, is pretty crude, very rigid, really can't get at mechanisms. Nevertheless, we had hundreds of peer-reviewed articles based using these BG methods. And it actually produced one of the first gene-environment interactions. This is an article by David Rowe, 1999. And it's, I think it might be one of the first with at least social demographic data that explores the SES gene environment. And here he is looking at, he calls it vocabulary IQ. This is a result of a PPV, a Peabody. Actually, it's a picture vocabulary score. It's an abridged Peabody, so it's not exactly the whole one, which is really important to note. The environment was parental education, again, fairly crude. And what you see here basically is that, as parental education increases, we see that there is a greater role of genetics compared to environment. But I guess most importantly for policy perspectives is at the low end of SES, the environment is more important. So everybody celebrated this. This means that there is a role for intervention. Right? So we were, I think, one of the first socio-demographic studies to actually collect DNA in a national field setting in the United States. So what we did at Wave 2 was we sort of made our own DNA kit collection, and it was primarily to check for the zygosity of some of our twin pairs. We tested for 89 twin pairs and clarified that. The way that we were determining the difference between MZ and DC, of course, were the standard survey approaches. And we found that there were actually MZ twins who thought they were DZs by their parents and brought up that way. Created a wonderful experimental design that Dalton actually has exploited. Happy to see that. Then we collected molecular data in 2001. This was Wave 3. And we collected it just on the twins and the full SIBs. At this time in 2000, there were no really commercial collections. So again, we used our own kit using a sort of mouthwash method for buccal cell DNA. We genotyped. There's data collection and then there's the genotyping, and we just genotyped some of the candidate loci in the dopamine and the serotonin pathways. We released this data in 2003, and it really generated a lot of interest, and it was some of the first, genetic analysis using molecular data, probably resulted in about 500 or more articles. Now, I thought I'd maybe talk a little bit about the challenges here, and then I'll go on with some of the evolution, because I actually want to get to the bottom of this list. So some of the challenges, some of the theoretical motivation, this is, I think that sometimes that gets lost. There's been some reference to this. When you design a study, and especially a study on adolescence, and it's about development, and you're collecting biological data, it's really important to motivate, why you need, why it matters for adolescent development that we capture the biological mechanism. Now, I know I'm kind of speaking to the choir here, but there was a lot of questions as to why we would do that and why we would want to spend, $2 million from NIH to capture this. So I wanted to, put this out. And I also think that as the methods for the collection of biological data become more accessible and available, a lot of times the reason for collecting the biological data is because we can. And so I think that it's really important, from the outset in terms of design to have that motivation. And so data collection methods and innovations. This is a very exciting area. We interview our respondents in their home. But if we can't get them in the home, we'll interview them at work, at the library, at the McDonald's. So the availability to collect biological specimens through Zoom methods is terrific. But a lot of times we're really pushing the, sort of pushing the boundaries on this, and we're using innovations that don't work. And in fact, that's probably more the story than the ones that work. And I can give you examples, but let me, you can ask me questions about this later. Logistics, very challenging. We're in all states of the U.S., Hawaii and Alaska, and we're a large study. So coordinating the data and the needs for testing it. We spend, probably just hours and hours and hours dealing with the logistics. An example, actually, I should show you something later on, but we collected urine and saliva to test for STI, or we collected urine to test for STIs at wave 3, saliva to test for HIV, but urine has a 48-hour window when the sample has to be returned, and so, and they would be too old, or the FDA test, would not be legit. So we were in the field during 9/11, so all those samples were lost. The samples that come from Alaska and Hawaii pretty much are not usable. Just some of those things. Lab culture, it's a really different, there's a different perspective on what data are in labs. And so there often isn't an appreciation for the fact that that assay result is linked to 20 years of data on an individual, and so the sometimes cavalier treatment isn't we're not on the same page. We've gotten lab results where there's been copying and pasting of Excel spreadsheets, and they're off by one line, which means that we merge it with our respondents, and it's off by one line. So then, these are not the same. So it's, again, tremendous amount of time. Costs I'll talk a little bit about later. Dissemination of genetic data is always an issue, and that's a big one, but we'll talk about that now, but very, very costly. So the other two challenges, cross-disciplinary training and the use of genetic and social data, and I'm going to give you some examples of this. The field is still fairly new, and so that's a real challenge, especially for somebody that makes these data available. And then the quality of the research. So I want to get there. So I sort of think about this as the good, the bad, and the ugly. And I take a lot of criticism on these issues, so I think about this a lot. But we have social scientists with no genetic training who use our data. and especially the new data. So the candidate genes. The GWAS are a little bit different, I think. And then, of course, geneticists with no social science training. It really does go both ways. And they produce variable quality of data. There certainly are thoughtful, collaborative, interdisciplinary approaches, like those presented before me. But I think that the first data that we put out in the 2000s, that there were many social scientists using the candidate gene data without much training or thought. So, for example, here's some here's some examples. You may agree or not agree of some of what we would consider the good, especially of these early data. Ben and Jason's article that came out in PNAS about the structure of schools and how that moderates the role of genes and friendship choices. I really like the article that I did with Domingue, and some people made reference to, age interactions. That's nice, because it's related to, that's birth date. That's pretty exogenous. And we found these really interesting genetic age interactions that were associated with risk behavior that, could be explained by sort of the fact that whether or not that, the behavior was legal or sanctioned. And those are good, you may not agree. So bad, we have a lot of gene-environment interactions. In fact, a lot of the examples I have here that have come out, and this is an interaction by my colleague Domingue that shows that number of partners is actually, is related to actually the school context in terms of the proportion of adolescents in the school have had sex by age 16. And so this, the 9R9R genotype is protective until the prevalence gets really high. So there are tons of these where the E is locally endogenous. And I tried to find the one with number of dinners, which was the environment exposure before, but I couldn't find that. And then, of course, there's the ugly. So we have actually a user, and I'm not sure if, this is all public, it's on our website. I felt a little bit bad revealing his name, but maybe you should forget this afterward. But Kevin Beaver is actually a criminologist at Florida State University, and he has 98 publications using adult genetic data. And here I just have some of the news headlines on this. So we found a genetic effect for belonging to a gang, having delinquent peers, and then the last finding basically has a quote that the way you parent a child is not going to have a detectable effect on their IQ as long as the parenting is in the normal bounds. So I've actually thought a lot. I've had users tell me you have to stop giving data to Kevin Beaver. That's the and we've thought about this a lot, and I worried a lot about this, and how I could because That Health has a philosophy of very open access to data. That was one of our promises to NICHD, and we promoted the use of the data, and we're very successful, and it's probably why we are still funded today. I finally came to the way that I look at this is that I feel like in the early years when the candidate gene data were becoming available to social scientists, behavioral science, this was a sort of a nascent stage, for the integration of genetics with social science data, and that there's probably going to be a lot of bad research in this early stage, and you have to go through that to get to the next level. And actually, I think that that's true. I don't know. Well, this was 2014. This was the last one, so I guess that hasn't stopped. Anyway, it's my life. So keep going. Wave 4. That was the most recent wave. We collected DNA from the full sample. That was about 15,000, and we use Orgy. Now there's commercial kits available. You can collect it, self-collect, send it in by mail. So this technology has expanded tremendously. What this looked like to us at first was that it was going to provide these great opportunities for replication. A lot of things that were out there. What we did in terms of the program project was we just repeated the candidate genes. And we wrote some additional grants to get GWAS data. So what I could say about the replication, and of course it won't surprise anybody, is it mostly failed. And then of course when it didn't replicate, it was hard to publish. But it did generate incredible interest. So when the first DNA data came back on wave 4, among our team of researchers, I had to really hope, everybody was just, rallying to do as many replications as possible. But for the most part, they didn't work out. So here is a site of a replication that we did get funded. That we did get published. And of course, this is the infamous CASB interaction. What we found was that we had the main effect of maltreatment. We tried to match this as close as possible to the Dunedin sample in terms of the age range. So we use a full sample. We tried to match as closely as possible to the maltreatment. And of course, it's not perfect. But we did not replicate the interaction or any main effect, which we didn't have anyway. But nevertheless, it had tremendous interest. Everybody was reading it. And we did look at it for blacks as well. And so that was a contribution. In some of the post hoc analysis, we would find effects, but they did not meet the multiple testing criteria. Okay, so back to the challenges. So the logistics and the costs are really high when your sample's big. So everybody wants a big sample. You're collecting data. So we always talk to labs and say, we're in the field for 1 year. We're going after 20,000 people. At the peak of sample collection, your lab may receive 500 samples a day. Can you handle it? Oh yeah, we can handle it. Oh yeah, no problem. We can handle it. Well, here's the picture that the lab sent back to me. Came in on Monday. There's probably 100 samples here. This is DNA, and they can't handle it. They've got to open every single one of those and use go through their system. Anyway, so when your sample's big, the costs are big. So DNA collection in wave 4, and then the genotyping, and we only genotyped like 10 loci, cost $2.5 million. Now, in the context of the whole grant, was $34 million. This is a large sample. At least we're going after a large sample, about 80%. The program project grant, I thought I'd write, is 800 pages. Just wanted to put that up there since I wrote most of it. I remember my predecessor, I said, well, how am I going to do this? And he said, oh yeah, don't worry, I'll help you. Yeah, no help there. Anyway, it's nice to actually say that to somebody. The GWAS then, we wrote subsequent grants. We had 2 grants. So Jason Boardman, Matt McQueen, and I wrote a grant for GWAS on the pair sample, so the original twins and full sieves. And then I wrote a grant for the rest of the sample. We had a little money left over to sort of start on the GWAS. Melinda was talking about 4 years. I don't know how long it's been, but we needed a grant for 5 years, so we had to spread the costs over those years. And so the total cost, again, is $4.2 million. And we're only talking about 12,000 cases. And I think at the time when I negotiated the price for the chip, so we started with the OmniQuad chip, which was $350. At the time it was like NIH was charging $500. So I thought I really got a good price. But then it took 4 years. So, and then of course in those 4 years, that chip was discontinued. So we had to use the new chip, which was the 2.5 Omni, which is great to get more markers. But then, of course, the price that went up. At the end, it was about $250. So, these are some of the challenges. Okay. All right. So let me end with a couple of comments here, and I think maybe some of these are really more points for discussion. What do we care about? These are the 3 issues I put up here, but I'm sure we can add many others. Care about environments and social experiences. So very attracted to the gene environment interactions. I actually am more interested in the gene environment correlation. So I really care about those environmental effects. Mechanisms, great examples that are usually missing in, I think, most of this research. So we're not so interested in the main effects, but biological pathways, linking the social exposure to phenotypes and causal effects. The endogeneity of E, as Dalton talks about, and the policy implications. I think those are that's where, the vision of making these data available, that's where that's coming from. Okay. Now, what do geneticists care about, and then do we care what they care about? And I would say yes. How do we attract them to our line of research? I worry there's not enough crosstalk between geneticists and social behavioral scientists. It certainly is getting better. I mean, computational biologists, for example, that I talk to at UNC, they have no idea what social science research data is and why should they? So I'm worried that we're losing sight of where geneticists are going and the importance of cross-training. Some of the current limitations, most of the GWAS are based on all-white samples. Of course, this is changing. The huge environmental variation, then, of the individuals is sort of just averaged across all of these environments. And then the polygenic scores, of course, come out of this. And again, we just really worry because, some of those SNPs, have, upward, downward effects, and they average out. The atheoretical approach, we have in demography, we used to always say that, if you, Dan sort of talked about the bottom up. If you torture your data, it will speak, and I worry about that. It's we've got the good statistical tools. Maybe too much focus on discovery and this notion that that the large sample is going to solve all our problems. I think that we need more diverse samples for GWAS. I didn't talk at all about sort of the measures of phenotype data. Adult is kind of a bad example in terms of specificity and sensitivity in terms of our phenotype measures because it's an omnibus study. And that's why it appeals to so many different researchers from different disciplines. But it doesn't always go so deep. We've got some, I think depression, cardiovascular disease, certainly obesity and BMI are pretty good. We need better characterization of the biological pathways, rather than focus on the discovery, but characterize just the way that the two examples. And then, of course, the genetic effects must vary by age. Most of the GWAS, I really don't, a lot of these are just based on older samples, and I'm just so curious, and so AdHealth, is about to contribute to that. But they, these relationships have got to vary by age, unless at least the biological pathways. Pay attention to measurement error. This sort of false idea that if you increase the sample size, it's going to take care of error. Wrong, right? It just exaggerates it. And returning to scientific. And so this is where I will end. I don't know if I was supposed to talk more about AdHealth. I'm happy to answer any questions, but I think some of the most compelling areas for social scientists is really in this, in the social genomics and the epigenetics, because it emphasizes the social experience, the exposures, and it gets at the mechanisms. What we're doing, the GWAS data is going to be in dbGaP in June, and it looks like we have about 10,000 cases that we'll be able. We're still actually finishing up some final QC where issues with the lab didn't match up. People don't always match up who they say they are. We have we've done the imputation. We actually have a grant. We got funded to produce transcriptome data, so we're really excited about that. But we're collecting patch gene samples on the entire sample at wave 5. So whatever that number is going to be, but we hope it would be about 14, 15,000. But we can only afford to process about 5,000 of them, which of course will be the biggest maybe transcriptome data around. I'm not sure. We have a grant in progress for methylation. Again, we can only afford to process 5,000. Afford being coming under a cap at NIH. So advice to me, please let me know. I feel like we're always chasing the next shiny thing. When I announced that at one of our advisory meetings that we are going to, we're going to do GWAS genotyping. Just got the grant. The director of NICHD, Alan Guttmacher, a geneticist, said to me, "Why? And that's because, that was now passé. And of course, it took us 4 years then past that point to even get the data. And then, the next shiny thing, epigenetics, telomeres, now that's maybe been a little disappointing. It's hard to keep up with the technology, and the funding climate is pretty tough, so I'll end there.
Question?
Yeah.
You had a slide there which said adolescents. We chose pairs of adolescents who were biologically related. How related? What is that?
Yeah.
mean exactly?
So, what I meant is that we have MZ twins, DZ twins, and then we have full sibs, and then we have half sibs.
Okay, so some sort of siblings. That's what you mean by it.
Yeah, that's what
Okay.
I meant. Yeah, but I
Okay.
sort of said varying. And then we have adolescents who have no biological resemblance, but grew up in the same household.
I see.
And there's 3,000 pairs of them. So we've got about 700 twins, at least originally.
Okay, just one more question. Is that initially you had high school, then filter, and then, so what was the filter in the graph which you showed? What filter?
Yeah, I went really quickly through those. Just
Okay.
But
Sorry, it may not be that relevant.
So you mean from the school base?
You showed the chart, the first few slides. I don't recall what they were.
Yeah, let me go. So I think the filter was.
Where you showed how you constructed this the data set essentially.
So we started with the school-based design. We picked 80 schools, 80 high schools. From across the country, and then a matching middle or feeder school. So this one.
Yeah, so feeder, not filter, sorry.
Oh, okay, yeah. So we end up with 80 pairs of schools, and then we went in and we interviewed everybody in those schools on a particular school day. So it's like a school census, and that gives us school context data.
I see.
But then we sampled from this 100,000 to come up with about 20,000. Dalton, and then I'll come. Yeah.
This is just I don't know if you can answer this question, but ad health seems to me to be especially now that you're going to have the full sample, have the GWAS data. It seems to me to be really, I'm trying to get the PSID genotype. But after the PSID, really the best nationally representative U.S. sample, because HRS has an issue that we'll hear about later about you got to survive a certain level to get into HRS. But this year's 50 years gathering them before they die.
Right.
We
only had 300 die.
So that's all the preamble to my question, which is I'm not aware of. In ad health or in other places, one of the things that I think is a big promise for social genetics, and that is deep history of reproduction. And I was recently, because I'm trying to basically I spend a lot of time, as I'm sure we all do, defending this why do we interest in genetics to train the social science to my colleagues. And I feel like one of the best defenses is to look at social history through the genome. So and I looked for examples of papers to, for example, look at X and Y chromosome imbalance compared to autosomes in terms of, look, you can tell the history of, rape and pillage, for lack of the delicate, for a better term, introgression of genomes that are imbalanced for men and women. I found a great example of analysis of mestizos in Ecuador, but I couldn't find literature on the U.S., for example, which, is that because ad health is really the only resource that I suppose we could do it with HRS, but it's really sociologists at least are not doing this, and I'm shocked. Are you aware of anything, or is that in your grant, or it's not really a question? It's just a flailing.
I'm not aware. The only other young, data set is Cardia, which is not representative. It's in 3 cities, but that's got a younger sort of age, right? But even for that, don't you? One time we were dreaming, especially when the pay lines are a little higher, is about, genotype, we're following up the parents and, getting the parents and having trio, at least for a subset. I mean, wouldn't you need that information?
No, I don't.
Right.
I love pedigree design for my own other purposes, but I think just knowing,
Just on the,
so for example, we know that PCs track onto from your demography article with Plum, but we can. I don't know. I don't know the sample size requirement, but I think with the new 10,000, you should be able to at least see, for example, PC1, or you're using admixture on race to see how much reproduction between blacks and whites or within the Hispanic population was European introgression from the male side versus the female side. I mean, these are really interesting questions that I'm surprised sociologists and or genetic historians, if there's such a field now, are not pursuing, because they seem less. I mean, when I describe this to sociologists, what we're doing, that's the part that I can defend the easiest. That gets away from like genetic determinism, or like why are you don't know the mechanisms for educational attainment. It could just all be through skin tone and height, and it's based on bias that's illegitimate. So why are you even investigating this? So, but appealing to like, okay, well, this is a record of who reproduced with whom throughout history. That seems to me they like that, but we're not doing that.
I don't.
As far as I know, again, I just did a literature search recently to look for this stuff.
I don't know of others doing that, and no one has said, I can't wait for you to get those data so that I can do that, which is what I want to know.
I was just, following on from that point, I was thinking that if you look at public support for ancestry studies, it's huge. We had a symposium. We have a Center for Evolutionary and Human Genomics at Stanford, and we had Christine Kennelly come down and talk to us, and she was talking about, she has this book, very nice book, about human history as revealed by the genome, and she was talking about Thomas Jefferson and that whole thing, which was eventually settled by genomic analysis. And I thought, if one could tap into a company like Ancestry.com or something, and we maybe set up some partnerships with them, then you could go after questions like that.
Yeah.
I mean, the sampling frame is really going to have to be different. But,
right. No, I think that's great. I was going. There's interest in building this large sort of administrative data that links across censuses and then taps into other administrative data. I went to a meeting last month on this, because we've been approached. Part of the motivation for doing something like this would be the availability to then link to, so actually have identifying information across census would be then to link to some of these ongoing surveys, and that would also help with the history, I think. Melinda?
I was going to ask you a question about what Dalton said about the U.S. data. So we've got a pretty good overview, I think now, at least but a lot of it's medical data, and then it's missing, So there is actually quite a bit of genetic data, but it's missing the social science part. It's really frustrating. But everybody measures age at first birth and number of children ever born,
Right.
Luckily, but. The question, or the point I wanted to make, you were talking about the beaver studies and these gang genes and things. And so I'm on the data access committee, the MetaDAC committee for the UK, which they decided to put all like the 1958 birth cohort, Understanding Society, UK Biobank under one committee. And the first thing they did was ban any studies on criminal behavior, homosexuality, and intelligence.
Oh.
So I just say that because that they just didn't want the data to be used for those reasons.
Yeah.
As a result, people looking at educational attainment and things, they actually had quite a few problems trying to argue why they, could look at the data. So, but people will still probably do it when they get the data, but at least at the at that level they've banned it.
Because we we were thinking about, when we put the data on DB Gap, we're not putting any phenotype data. I mean, I think height and some of the sampling design parameters. And then once approval through DB Gap, then they come back to us, and we have, just our basic data contract that everybody goes through for restricted data with a few additional. And then then the question was, well, then we just give them the link, and they have access to the whole data. And it's, I mentioned cost. It's an issue that we are always thinking about because it just costs us more, to customize in any way. But we were thinking, well, maybe we would take out the criminal data. I mean, I don't know, but it's, it's
hard
fuzzy.
to take out the variable of that has labor market status usually, or do you know what I mean? And then,
exactly.
and then so you take out their activity status variable because they're institutionalized. So
Right,
we
it's
sat
just-
there looking at, you're taking out some main variables when you do it.
So one thing fascinating in those lines you pointed was they seemed rather causal statements more than correlational statements. Is this very typical of studies like this, or is it not?
Some of the examples that I...
Yeah,
Bad science. This is bad science journalism.
Yeah.
Which is especially
okay.
Yeah.
Because you could find a correlation for a bunch of reasons. This doesn't.
Absolutely. I mean, I think that, you have this belief in, at least we did, I don't anymore, in the peer review process. And that was one of the things that we, sort of rested on. But there is a peer review process for publication, and then there's a peer review process for the evaluation of good science. So, yeah. But I mean, I think that it is. Well, I don't know. I mean, any others thoughts, about that? Be sure to pass them on to me.
Okay, thank you.
I've just been asked sort of discuss the now beautifully demonized sort of biomedical science sort of view of all of these issues, and you've set me up fantastically, Kathleen. Could not have been better to talk about it. So what I want to do is just talk for a few minutes, sort of kind of placing where I think the biomedical community, from the ugly to the best, are thinking about these issues in relationship to the broader agenda that's evident in the room. And then I want to spend a couple of minutes talking about the data explosion, because I actually have a slightly different spin on the last little bit of discussion here about what's potentially available and among survey data, biomedically generated data, and administrative data. I'll just sort of betray my spoiler here, which is I think we're actually in better shape than we think we are, and we're heading in an interesting direction that's going to allow for some pretty exciting work to go on in the future. And then, depending on where we are after those couple of minutes, I may or may not talk about one particular area that you're going to hear more about tomorrow, namely Mendelian randomization, and the impact of the explosion of linked data sets for the possibility that social scientists may inherit from the biology side some new tools for causal inference. And I'm not going to spoil the thunder of a great talk you're going to hear tomorrow, but just talk about the conception of that, which I think has gotten a whole new life. But anyway, let me start by telling you a little anecdote, and I hadn't even thought to tell it, but yesterday there was a big data conference at Stanford. We have them a lot, and because it's a real exciting area. But in the run-up to it, there was a by invitation only meeting with Claudia Williams, who's the White House liaison for the Precision Medicine Initiative. So she herself, not a geneticist or a physician or a social scientist, but more of a political liaison to the science community. And invited to the meeting were folks primarily, at least from the inside, folks from the medical school, geneticists, cancer folks, folks in some of the informaticians who've been interested in looking at and strategies, bioinformatics strategies for causal inference in biomedical areas. But also invited were some venture capitalists, some folks from the tech community, some folks from the pharma community. And Claudia started the conversation by just describing the process the White House is going through and trying to very rapidly roll out this enrollment of 1 million volunteers who will have extensive biologic sampling done, as well as ultimately hopefully linking their electronic medical records to that, a subject I'll come back to in a few minutes. But after she finished this and so forth, I asked her, I took advantage of asking the first question because I suspected correctly that it would be my last opportunity to ask it, given the direction the discussion was going. I said, so, one of the things that was not clear, because we actually had bid on some of the initial RFAs to play various functional roles in this process, partly because we wanted to be able to control the data. If we could, we lost. But one of the things I personally had given a lot of attention to was the extent of environmental, social, and related data that you all would desperately want as part of a great trove that was developing. And so I just said, what work is going on, and what's the process going to be for deciding on what that portion of the data vault is going to look like, and how you're going to collect it, and what you're going to collect, and so forth. And she looked at me, and with all sincerity, she said, look, we need to walk before we can run. And by that, she meant we need to figure out the biology. We need to figure out how we're going to recruit the people and draw their blood. And that was walking, and then running was actually trying to think about what we might append to all that. And of course, I was hardly surprised. I was a little disappointed, but I was hardly surprised that that's where they are in their thinking. But it underscores what I'm going to now take you through is sort of a progression of 3 ways that people in the biomedical world are thinking about genetic and related biological data. So the first that are people that are into precision medicine, which is actually a good word for describing what's actually been going on for 20 years, long before we had whole genome sequenced, when we were still looking at single gene studies and so forth. The idea was to identify variants that were actionable, and actionable meant, frankly, a drug, a therapy, a treatment, usually a very expensive one because it was very targeted and required a lot of technology to devise, and as the technology to do that got better and better, and God knows it's incredible, and if you're one of the unfortunate people that has one of these weird diseases, you'll be very thankful that we can do the things that we can do, but that's where they're going. And frankly, that group took over the conversation yesterday in 2 minutes. So it was only the pharma people and the geneticists and the cancer people, because one whole sideshow of no relevance whatsoever to anyone in the room, unless you're coming from the biomedical side, is the extraordinary success, well, sort of success, that they've had in cancer, where they're doing the genetic somatic mutation. They're basically looking at the genetics of tumors, and have discovered that if you actually can code the tumors, you can classify them into treatment groups and do a bit better. No remarkable big successes just yet. Well, there are a few, but I mean, it's not magic. But it's a great and fantastic thing. But that's kind of what precision medicine is turning out to mean. But as I said, I'm going to come to a happier part of the story in a second. So then there is, Sardine got this idea, a little unhappy with this, and too much to his credit, of the notion of precision health, in which he sort of incorporated the fantasy that 23andMe is introduced in our society, which is sort of the predictive notion about of genetics. And you're now not, the commercial aspect here is the 23andMe. So you can sell people expensive kits, and people that have the money and are curious and want to know something a little bit about their ancestry and a little bit about what their risks for bad health may be, are opting in. 23andMe has sold 1.4 million kits. Don't underestimate how much profit there is to be made. There's a whole lot of direct-to-consumer marketing. Very few physicians have actually gotten the business of ordering such tests directly, other than very specific ones for where there's a family history of one or another dangerous hereditary disease that you really do want to pick up early in life. But it's a huge thing. But to his credit, the notion that forget the value of this to expensive therapeutics. Think about what we might learn from the biology that would allow us to more personally and in a more precise way think about people's long-term risks, and maybe carve the recommendations one might give them in a somewhat more personalized way, based other than the traditional ways we've thought about health risk going forward. Now, between you and me, the margins are pretty small. So, for example, if you take all the risk scores for things like diabetes and hypertension and cardiovascular disease, Alzheimer's disease, and so forth, they tend, collectively, they predict less than we can predict from 3 questions in any of those spaces so far. I mean, that doesn't mean that a lot of the unexplained variants may show up someday. We'll talk about that. I'm very hopeful that it does show up someday. I don't think it's going to show up on the unexplored part of the genome by itself. But there is still the fantasy of one more shiny object idea, one more fancy thing. When first they had SNPs, they said, oh, it's going to be in the full sequencing. Now they've sequenced the exomes. They still haven't found it. And so now they talk about gene environment. But of course, they talk about gene environment exactly the opposite way do. They view the environment are those things that need to be known, not because we're interested in studying or changing the environment or the way people behave or things like that, but because that'll allow us to see how strong a predictor these these genes are, and to better kind of understand where they're going mechanistically. But of course, I don't think any of us in the room have to be satisfied with that, with either of those paradigms as particularly interesting at least from an intellectual point of view in terms of our understanding human health over the life course. Because the reality is that the tools, these very tools that are going to make $1 billion for a lot of the people sitting in the room yesterday, are actually tools that have phenomenal potential, if we got the right data sets and thought about them in the right way and solve some of the analytic problems, which are beyond my expertise, but there's some incredibly smart people in the room that are thoughtful about this. We actually really could begin to sort of see the life course, and I'm going to lay it out one funny way. So I'm out of the evolutionary context right now, although I'm fascinated by it. But let's start at what most biomedical people think is the beginning of time 0, which is the moment of conception when you get your endowment, and ask the question, so now what happens? Now we'd have to think about, about intrauterine life and then 80, 90, if you're lucky, years of life. What is day by day, step by step, behavior by behavior, environmental exposure by environmental exposure, social context, social environment by social environment through the various critical stages of life? What are they contributing? How are they modifying that original endowment to lead to states of health and disease? And the fact of the matter is that, putting all cynicism aside about who's going to benefit from what, we who actually care about many of those elements now, for the first time in theory, have the opportunity of watching the whole thing unfold. So, for example, how, for years people have wondered sort of how do all of those social things get under the skin and change our health. And for a long time, as for those of you who've thought about health disparities or you sort of watch the literature evolve on the emergence of health disparities, there's been this notion that the brain is a target organ, and what used to be thought as homeostatic responses between the brain and the inflammatory system and endocrine system was kind of viewed historically. When I was in medical school, they taught me it's homeostasis, and so the new notion was, no, it's not homeostasis. It's an allostatic process which is changing with use. So as the brain is bombarded by various environmental stressors, writ large, it's changing in its response. So it deals with the short term, but at the price of the long term. And it's a really attractive theory that Bruce McEwen and others who've developed it, very highly touted, New England Journal, PNAS type stuff, in all the neurojournals, sort of, this has been the paradigm for sort of that part of life. But of course, most people were sort of unnerved, I was certainly one of them, having worked a bit with Bruce, that you really couldn't, in a 20-year-old or a 30-year-old or even a 10-year-old, easily demonstrate that allostatic change in a way that correlated with any observable changes in health status and so forth. And the evidence that the brain was where the action was occurring was not great. And then along about 15 years ago with Elizabeth Blackburn's work across the bay and Alyssa Pell and others, the whole idea of telomeres popped up. And oh my God, so maybe the brain is sort of translating the environment for us and changing, interacting with the cellular reproductive mechanism in such a way that telomere length was progressively shortening, and that may be the scorekeeping Because after all, I mean, diseases don't happen from stress. I mean, stress doesn't obviously lead to biologic change. I mean, we need to sort of understand biologically how all the things that you all in the room are actually interested in as phenomena actually do it. And now, for the first time, we actually probably have a glimmering, maybe a good glimmering, of what's going on. So here at the epithelial level, the cell level where the diseases are actually occurring, some exogenous things, physical environment's easy to understand. A lot of the aspects of behavior are pretty easy to understand, how they might interact with cellular function. Social environment is still a little of a mystery, but imagine the brain is processing that information and signaling our epithelial cells in some way. But we now have at least some pathway by which the genetic endowment may be modified in its expression, leading progressively to changes that we probably now, for the first time, can actually measure. So, the endowment is interesting, but its interaction with the environment is even more interesting. And we probably, if we get the right data sets, which I'll come to in a sec, will have the opportunity of watching as each stage of our life either accentuates good opportunity and accentuates the best of the endowment that we may get, or the reverse. And unfortunately, for most of us, it's kind of the reverse that we fear. But at least we now have like a paradigm that I don't think 10 years ago we really had. It's way different from the paradigm that precision medicine people have who are looking for targets, although many of their tools are going to be fantastic. Because we actually do want to know at the molecular level what those mechanisms are, not because we're looking for $1 billion drug, but because if we're going to think about social forces that can really make a difference, we need to know where to interact and what the impact of that's going to be. And so, so we're actually aligned with the biomedical community in trying to better understand those mechanisms. We just have a completely different takeaway. I mean, our focus of that is different. So anyway, so let me just swing for a second to the data side before I just open it up for your thoughts. So, for years we have been beholden to folks like HRS and its counterparts and PSID and AD and other fantastically expensive, well-designed, thoughtful studies to give us the kind of social information that would allow us to study the relationship between health and the way we live. Every one of those studies omitted some broad area. I, for one, am very interested in physical environment. And so in most of these studies, HRS, even though I would think it's sort of scary in retrospect, but I was called in the late 80s to help design the occupational and environmental parts of that, and we failed to do a good job in HRS. Many other studies, I think, have also failed to figure out how to do it in a general population way. But we can now measure all those things. We can, all these same little toys. We laugh at the Fitbits and all those kinds of things, but we're literally a couple of years away from being able to measure people's physical environmental exposure, maybe a couple more years before we can translate food consumption into that level of analysis. So frankly, in terms of all of the environmental factors, behaviors are going to be easily measured, physical environment is going to be easily measured, obviously the socioeconomic sorts of things and social interactions, that's going to be much harder and require a different way of getting that data, and it may well require the continued use of dense survey techniques. But for the rest of the what my genetic friends call metadata, it's like the word that just sends chills up my. Metadata is the few tags of information they need to do GWAS. You're diabetic, good. You're 53, you're white, perfect. I say it laughably because it's laughable. I mean, it's, and when you have to put 63 of them together, you see where you get to the pretty low common denominator, what the metadata looks like. But that's ancient history because nowadays we have incredible troves of information. So let's just start with the obvious. So even though they're not interoperable and they're a mess and not everyone has one and so forth, but by and large, most of people's medical histories are now digitized. And for very large swaths of the population, you go be shocked next time you show up at some hospital you've never been to before, how quickly as you roll into the ER, they've got your life history. And you look and say, where the hell did you get that? It's because health information exchanges are going on, and much of even the ancient stuff that wasn't digitized is at least now scanned in, and much of it's been mined to fill structural data. So electronic medical records, which of course they're way more useful to people who are answering biomedical questions than they would necessarily be to social scientists, but there's a lot in there. I mean, someone commented that they have gravity and parity and all those kind of good things. Yeah, they do. And they probably have a lot of other environmental data, like people's drug history and a lot of things. Is it perfect? No. But compared to surveys, it's not so bad. A lot of these things are now linkable either geospatially or through other forms to administrative data. Don't underestimate administrative data. No, we're not Denmark yet. We don't have everything all linked through a national identifier. We have huge privacy problems, which are going to, just screw up a lot of our best science, unfortunately, because we're going to be stuck with voluntarism and selection, endogenous problems that are going to make causal inference difficult in a lot of the best data sets we get. But nonetheless, the trove of stuff that in the federal RDC, the census troves, and there's a hell of a lot more data out there. We've got Social Security, and we have IRS, and their life stories. The IRS has your life story. For those of you who don't know it, the Census Bureau just released, per Roosevelt's ambition in the late 30s, they released the first batch of census reports of people over the age of 72. Now it's been completely digitized. You can get a file, we have it, of every American who ever lived in the United States prior to 1940. You can get their decennial census surveys, which means for someone born before 1940 that you can find their home. It's easier for men than for women because the name changes. It's matched by name. You can find the home they grew up in. You can figure out how many square feet it was, whether they owned it or rented it, what the parental occupations were, what the household income was, how many sibs there were in the family, an incredible amount of information about social history stuck in those census forms. You can get it online. Go look for your father or your grandfather. It's there. If they were in the United States in 1940 in their own handwriting, you can get the original. But it's also digital, which means we can start linking those things and learn a lot about children in cohorts that we have followed. In fact, we're about to now look in the Women's Health Initiative and in the Alcoa study, which has been my child for a long time. We're now about to start look at the people in our large cohort who are over the age of 72 and very, very precisely nail down their childhood circumstances through this information. Pretty amazing. And frankly, this is the way it's now going. So you may laugh at the Precision Medicine Initiative and say, oh, God, they haven't thought about it. There are terrible limitations on the UK biobank still in terms of what's linked. But if we're compelling in our efforts from the population science point of view to make the case that as much that everything should be structured in a way that it's linkable to administrative sets, and every data trove that's built that can't be directly linked to those sets the biggest effort possible should be made to collect the social information that is otherwise missing. I think we're actually in for better times, and I think we are going to start having data sets, even ones that you would just not even think about, because they were designed so differently and with such different purposes. Like the million vets, like the million people from this, like the 500,000 in the UK, that will have a ton of linked data, and we should be the first ones in. We should be mining that data, religiously to try and pull out the stuff that will allow us to put together the life course and fill in all these gaps that are so critical to us, whether you're interested in aging and retirement, whether you're interested in early child development, whether you're interested in the transition from adolescence to adult. A lot of the goodies are there. So I'll stop there.
I just want to build off of a little bit of what you're saying about reaching out to the biomedical community, maybe add a little bit of context to that. So within hospital systems across the country, because of the Affordable Care Act, two big things are happening. One is that everyone has pretty much had to switch to electronic medical records, which obviously makes the medical data much more available. The other reason to get involved is that is the Affordable Care Act's sort of subtle pushing of healthcare systems toward forming ACOs. So for those of us who live in the Bay Area, you probably see hear radio advertisements for Stanford Healthcare or UCSF or Kaiser because there's frankly a quite nasty fight among these healthcare systems to dominate the market and form an ACO, because that's the only way they'll survive. Those of you in North Carolina probably see something similar with UNC versus Duke, and the point is not that we should all come to fisticuffs, but that these systems are realizing that they need to treat their patient pools as sort of an audience that they follow from beginning to end in the electronic medical record where things get picked up. So one, this is great for us as a source of data, because now hospital systems are really going to follow people in and out of the hospital for longer periods of time. And two, and I think this is a really critical point, not only is it a great source of data for us, but it helps us justify what we do much more effectively, because if we're working with medical data, we can build collaborations directly with the medical schools and their associated healthcare systems, and not just talk about these interesting findings we have. But one criticism that I've always gotten about this work is, well, okay, so you found some gene-environment interaction. Well, what do we actually do about it? Because doctors are used to the idea of prescribing drugs, and we poo-poo behavioral interventions. But now that we actually follow patients outside of the hospital, behavioral interventions become much more important. Environmental interventions actually become financially the only way to go for some things for hospitals. And that's sort of what we know, the people, all of us in this room, are understanding behavior, studying behavior, and thinking about intervening on it. And suddenly it justifies our existence much better, which is nice. And it offers another interesting avenue to our research, which I don't think anyone here will be talking about, and I don't think I've ever seen discussed, which is intervention research. Okay, now we know something about the genes, and we know something about the social environment, we know something about how they interplay. Now we can design intervention studies that take advantage of that. And so, I guess in conclusion, reach out to your medical schools. Those of us in the UC system or who will be joining it, it's a great opportunity because all of the UC hospitals throughout the state are linking up their medical records.
No, they're linked.
Trying to really...
They're linked. It's 15 million. They're not perfect, but it's 15 million.
It's not perfect, but it's going to be one of the biggest in the country, and it'll be, from all the way from Davis down to San Diego. So this is a conversation. So we should be going across the bridge to bugging the people at UCSF who are working on this. I'll get off my soapbox for a second.
So I don't know if you're aware or not, but there are people who already mine massive quantities of data which you can purchase, and it wasn't exciting, it was horrifying. Anything you've ever purchased on Amazon is worth 20 cents, and they know it. So nothing is private about any part of your life, and it's for sale to anyone who wants to buy it. So, I mean, you'd be surprised at how much data you can find about a person, and they knew, well, you purchased a car, you purchased a Honda so-and-so, you purchased it in 2012, it was white. And that's too much information for anyone.
Well, so putting aside, as I said, privacy is a whole other serious discussion that I know bothers me. But this transactional data, this administrative transactional data, is stunningly informative if you actually want, for example, to reconstruct someone's lifestyle without a survey. It's
so to your point, it's available, and there are people who are willing to sell it to you, and I know the people, and it's legal apparently. So.
Linking it with data that you've acquired under some sort of IRB approval is a slightly different issue. So I think that's one of the barriers. The commercial sphere, nobody asks permission. Right. They're dealing in a sort of insufficiently regulated space for the time being. But for those of us who are on the other side of that line, reaching out and linking Amazon purchasing history to the medical record that I've obtained consent from the person to look at, I think is not not there yet.
I'm currently working on fraud. You don't know who the fraudster is. They can say a surprising amount about a person whom you don't know based on IP addresses. It's not data which is obvious. You're not going to say, my name is John Doe, give me information. They say it's surprising how much these fringe things tell you about a person. And there are patterns in fringe things too, like there are things with John Does who do these sort of things also do. And their purchase patterns say a lot of stuff. And their point was, so the people who gave the talk there said, we're ready to sell you, A, we're ready to sell you this information, B, we're ready to sell it to you on a monthly basis.
So let me bring it back down to earth, because that makes you nervous, and as I said, I don't want to. But how about this? So how about if you had access to
Safeway's purchasing information, so that you basically, for large portions of the state of California or Texas or states where they are major players, could reconstruct their weekly food purchases.
They already do.
I'm saying, I'm trying to say, would this be useful for doing various kinds of social epidemiologic research and trying to make sense of one huge exposure and behavior category that we otherwise ask people badly and get very, very low quality information about consumption? So, I mean, I have many of the same, anxieties about the scope of administrative data that's out there. But I remind you that these are the gaps that we currently have in trying to knit together the story of a life course. And for example, food's been one of the biggest problems. We talk about diet all the time, but it's very hard to study because we have such crude tools. Okay.
Well, my comment, I think that's a data quality issue with all types of data, so there's got to be a process. But the analysis of these data, linked, there's the IRB issue for those of us, who have contact with human subjects, it would be in these RDCs. For the most part, I think that would be the only, at least anything like that help, that would be the only access that would be allowed. So, what are people's perspectives on that? When I was approached, what we had to think about linking to the IRS and, the census data. The IRS data would, for example, would give us address, every year of our respondents' lives, which means we could create that both the built environment.
So they'll never give you
social
that.
environment.
They'll let you use the data for coefficients, but they're never going to give you their data. But if you want it, for example, if you need to know household income, they'll give you, they'll use it, but you'll get the beta. You won't get the individual household incomes.
I think the big plan is that it'll be linked. In other words, they're not just going to give that we would provide the identifiers on iHealth, and they would link it, and there would be a linked file to whatever whatever was asked for.
As I said, we've worked with them with both IRS and Social Security.
I'm talking about Skyify.
Yeah. No. Maybe someday.
Not now. But that would fill in. That would be, that would give you all the social environment as well as the built environment over the entire life of the athlete, and in the pre-conception period, which is, which people believe, it's before conception that those factors matter. So, but that could only be analyzed in an RDC. This is the point.
Yeah. No, I think that's possible, or something like it. No, I think it's probably true.
But the discussion we often have in the UK on these data access committees, everybody gets a consent forms out. So what did the people consent to in 1958, for example? The 1958 Britain, well, nothing we're doing now. So you get into this huge legal aspect of what can you connect and what did you say you were going to do with the data? And a lot of the medical data, and we've trying to get these cohorts. I figured out what the consent forms are for a lot of these, and that's a lot just for medical outcomes. So everything that we look at is often not related to medical outcomes. So you have to position your research as a medical outcome. And they didn't consent to have it linked with Amazon data, or
But one of the advantages of some of the new cohorts, like the precision medicine initiative in the States, is the breadth of the consent that they are actually asking people to provide. So that these are people who are really going in as health volunteers, and they know it, and that they will be subject over time to fairly extensive research done by many different groups, not just friendly groups, but by others who will have access to the data. But they're consenting, so they consent to a lot of those kinds of things up front, which is of course not true in a lot of the cohort studies in which the consent is different. Yeah, totally.
So two things that I wanted you to opine on. One is the idea of forming partnerships with profit-making entities. And I thought of this because David and I actually had a meeting with Johnson & Johnson. And one of the things the guy said to me, which I thought was really interesting, he said, we're a pharma company, but we want to become a wellness company. And this is a big thing across the entire pharma world. They all want to be wellness companies. But they don't know how to be wellness companies. And so they are very interested in partnerships of some kind. So that's one issue. what do you think about this, and how does it, is it likely to corrupt the science we do, or is it a way of opening doors? And the second question that I had related to this is I, have still not quite gotten out of the habit of looking at NIH opportunities for funding.
It's an old bad habit. Sooner or later they'll break you up.
They have. And one of the things I noticed is there are sections of NIH that actually do these intervention studies, where they ask you to go out and find populations and, conduct wellness studies and so on and so forth. But those people don't talk to any other part of NIH that I'm aware of. And I was thinking, surely, that would be something that, social scientists, biomedical people, and nurses and whatever could get together on. So.
They're great questions. I guess I'll just give a personal view of the first part of your question, which I think is answerable, which is that I don't think we should be so proud as to view our partners on the commercial side as unworthy partners. First of all, unfortunately, because they can afford to do it, they attract considerable talent, and down where we are in the peninsula, this is a steady concern for us, is that many of our, especially in some tech areas and economists and so forth, are constantly being pulled off in that direction. But they also, for reasons that are not wildly unrelated to questions that we have, they often have troves of information. So they may be using the information to try and understand on what portions of their population a drug may cause adverse reactions, or what the underpinnings of that are genetically for their own profit-making reasons. But it often leaves them with large troves of medical claims data or other EMR data or other things. They are some of the biggest repositories now. And again, putting aside some of the ethical issues having to do with the individual research context, to the extent that we need places to merge biologic, health, and social information together, these may be some of the platforms on which it can be most easily done. So it's hard to know.
I was wondering if you could comment for a minute on the issue of selection, both when we talk about consent and the fact that more and more as these consent forms, because more broadly are, as you say, sort of those that are consenting are really health volunteers, and also to maybe to less extent the electronic medical records. In some ways, there is a selection into health-seeking behavior.
For sure.
And to be clear, I think I agree with. I think this is kind of the way things are going to go and the way they should go. But the extent to which selection is a part of this new data paradigm seems like something worth discussing.
Yeah. No, I quite agree, and I've always been very nervous about any inferences we draw, partly because my background is in studying, cohorts like occupational cohorts that are complete, and I have a great, predilection towards geographically or geopolitically or otherwise defined populations in which selection is just not. There's selection, but it's not based on volunteering for one thing or another. So what the selection factors are, you can usually deal with. Oddly enough, our friends on the internet have figured out a way to deal with that problem, which is they make the consent form so long and arduous, and then if you're really smart, you can quickly click down 3 times and come to I accept, and you never have to read it, so you have no idea what. That's it. That's the origin of why all that stuff's available for sale, because none of us has any idea the last 700 consent forms we signed. Many of you probably signed one this morning without knowing it. There you were in Starbucks or something. Boom, I consent. Sure, let me have the internet for free, and I don't know what I told them. I'm sure they know when I go for coffee. Yeah, it's like that much information is clearly on the street. But we laugh at that, and from an IRB point of view, it's horrifying. But from a selection point of view, it's great because it's frankly it's such a big sieve that it's basically everybody. I'm being facetious. No, those selection forces are going to be hugely important. And again, I think most of us sitting in the room tend to be pretty sophisticated quantitatively because we think about the populations from which any sample were drawn. It's part of our innate training, but most of our friends on the biomedical side, think, one person, you've seen one person. And when they talk about GWAS, someone, I think, put it real nicely where they say, oh, we need 1 million people. Oh, let's get 1 million people. It doesn't matter whether it's all the people that live in a particular city or whether it's 1 million people who raise their hand, to say, I want to know about my genome. They say, one genome's the same as another. And, of course, we have good reasons to believe that's false. Yeah.
Yeah, I think to be pretty critical of that notion that the biologists basically are thinking in terms of fundamental biological processes, therefore representativeness from a social science perspective is not so important. Recently I've read the work of my colleague Sid Krishna and others that have suggested that convenience samples, not working in this area at all, working on sort of data exhaust and kind of Twitter and other big data domains, but really that with post-stratification adjustment, we can use convenience samples and we're not so bad off. Now that's a different domain, but maybe they're right in this area too, that we don't, as long as we know some basic demographics, take 5 or 6 variables and we can adjust our sample afterwards with weight. Why not just get
so I'm less interested in
or 23andMe or whatever these unrepresentative samples are. Of course, there's going to be some limited variance in terms of like educational attainment. I guess there's hardly any high school dropouts on 23andMe, I'm guessing. But that's something I've kind of shelved out in the back of my head. I think, well, maybe we make too much of a fetish in this day and age about representativeness. I have for sure, and we can deal with it other ways.
Well, you can harmonize, and you can come up with strategies with as we do with observational data, in which you sort of post hoc randomize instruments and things like that. In fact, one of the things I was going to just comment about briefly is that one of the phenomenally good things that's happened with a lot of these formally survey data, like HRS, is all of a sudden now they have a genome, which at least creates the opportunity to use Mendelian instruments, at least for some things. And you're going to hear a talk tomorrow in which I think this was very cleverly done, and creating a whole new strategy for dealing with all the selection pressures in large populations. It's not perfect, because I think you may not have ever thought about this, but Mendel was right about the first 2 principles, but didn't understand the degree to which environment was going to reflect on the non-randomness of the expression of all of those genes. He studied a very lucky environment in which that wasn't a problem, because all the damn beans grew. But we, of course, know otherwise in our environments. But I agree. There'll be strategies for dealing with those effects. We're going to have no choice. Those are the kind of populations we're going to get. Okay.
Well, I'm struck by your bringing up this topic because this meeting is one of a series of mini-conferences sponsored by the Berkeley Population Center, Center for Economics, Demography, and Aging, and the Stanford Population Center and Simons Institute. And the other topics in the last 3 of this series have indeed been on the subject that we're describing. The last meeting over at the Simons building was on, Matt Sopanek was there, and it was on network data and possibilities for big data control for selection biases and that interaction with network sciences. One of the things and they're making huge progress in, especially in political fields, with successful post-stratification reweighting of very strange convenient samples. But they're doing it with 15 million, 100 million, 500 million observations, so that there are all the cells that become significant for the selection process have occupants from the data set.
It's the crowding notion. It's basically the crowding notion. That if you have so many observations and you know so many things about everybody, is selection anymore? Do you have to worry about selection? What's left endogenous that you haven't measured?
So what you're describing, it's big data, it's sort of small big data compared to those worlds in terms of the samples and the occupancy of cells. I think it's going to be difficult to bridge unless there are much more open possibilities for data linkage, at least on the few of the variables that one can get, whether one can leverage, one can get post-stratification information by linking to very small numbers of variables in census or huge in this sort of data sets. It's going to be a very difficult problem, I think, in the rich data context that you're talking about, to get large enough samples to do that control.
No, I mean, I agree, although I think it's going to be our challenge is to try and figure out strategies for legitimately doing that. Yeah?
No, so there are sort of two issues going on with representation with this issue. One is consent. I just want to raise that, so Vanderbilt had a system like this, so they're one of the most advanced places in terms of bioinformatics and medical informatics in the U.S. And it used to be that it was opt-out consent. You went to a Vanderbilt clinic, and the assumption that you had to go under was, okay, my data are going to be used for research. If they draw blood from me and they have any left over after the lab test, they'll probably do something with it. And that flew for a number of years, and then in the past year or two, they were told that's not okay, and now it's opt-in consent. And interestingly, didn't drop participation all that much.
Yeah, it did. Fair enough. They're pretty nervous about it. They gave a
about
talk yesterday. They're pretty nervous.
Yeah, they're nervous about it. I got that impression too, but it's not as bad as anyone seemed to expect. And so I guess revisiting the issue of opt-out consent is something that we might have to keep doing going forward. Even in social science surveys, maybe the idea should be that there may be extensions to these data that will be useful to researchers. If you don't want us to extend your data in that way in the future, we can't anticipate this, then opt out of us extending it. That would be a way to go forward. And then this issue of representativeness in surveys. And I think we're talking about two different issues. There's representativeness and then there's representation. So there's representativeness, does it reflect the population at large? And we can adjust, we take NHANES, which is only, a few tens of thousands of people, and we adjust that regularly to reflect the U.S. population in the years from which the, source of that sample was drawn. And we seem to do it pretty successfully. At the very least, the epidemiology community believes the figures that come out of it. But what we're talking about often are disparities, between people of different levels of some measure. And there, it's not so much that we care about our sample being representative proportionally of everyone who's a high school dropout and or a college graduate, but just that we have these groups adequately represented in these samples. And I think if we view the problem that way, I think it becomes much more tractable. If taking 23andMe, for example, if the issue with 23andMe is you want, take your example, you want to look at high school dropouts, and you have a sample from 23andMe, where there's not going to be very many, the solution is, to me, somewhat straightforward, which is you go and you find a sampling frame of high school dropouts. How you find that frame, I'm going to leave as a problem to everyone else. But once you find that frame, you can then just get consent and pay them to do 23andMe, and then you can make your comparison happen. And this is sort of like chapter 2 of Rothman's epidemiology introduction. We don't necessarily want representativeness, but we do want representation.
Emil?
Well, I think I put my hand back down because I feel like I might just be repeating what I said 10 minutes ago. But I'll push back a little bit, which is to say that I think selection and representativeness are obviously two different issues. And so what I'm just doing is going back to selection. But what I get out of Dan Dan's talk this morning, and to some extent Melinda's talk, this idea that where we are, I think, in terms of this kind of G-by-E world, and especially when we use polygenic risk scores, is that we're capturing something that we are measuring as educational attainment, or as reading, or as I think Melinda, you said it was kind of a middle class marker or something, right? And we don't know if that's something or those some things are yet, and I think that's just where the science is, and there's no problem there. But the samples by which we have more or less of the people that have these some things in them, I think that part matters a lot, because I think when we think about effect sizes and sample sizes and kind of what is rising to the surface in terms of what we're learning, kind of who's coming into these samples to begin with, and the extent to which there are differentiations across these things that they have that lead to higher levels of education and other things. I think that's just, I don't think it's an intractable problem. I just think it's something we need to be really cognizant of as we grow our data to include a variety of sources.
To make this concrete, in longevity research, this is an extreme problem because Ben's going to give a talk about mortality selection tomorrow, so I'll leave that to you. Sorry. Feels like the distant future. But what determines variation in lifespan in individuals older than 50 and in individuals who are, let's say, 20, may be different. And the same may apply if we're thinking about what determines variation in economic attainment among individuals who are broadly middle class and well educated as compared to other populations. And so what we uncover may be tailored to the particular populations that we're studying in ways that we don't fully understand. In fact, this is, I think, a tremendous issue in mental health genetic research, where we've traditionally been studying individuals who not only are in treatment, but who are in treatment and volunteering to turn over their genomes. And that may be a fairly special population, particularly in the case of certain extreme disorders.
I think I agree with most of what you say. It's not, two things are not obvious to me. The first is, I'll wait for Ben to present this stuff.
Before you really start to slam it?
Before you really
No. I think it's great stuff. I think it's a very insightful way of trying to address the question. But it's not obvious to me that I think what you've done is you've found one possible answer, and it's not clear to me that there are not an infinity of answers. So that's one issue, and you can convince me otherwise. But I think that two other points about that. One point which I think it's not clear to me that is part of the analysis is in the case of mortality selection. You can look at variances between subgroups, and that may actually be informative in that regard. So I think if you look at the variance in age of death between high school dropouts and people with a college education, as Ryan demonstrated a long time ago, it's about the same. So if the question is, should we make sure that everybody gets a PhD in order to live longer? The answer is, on average, they'll live a year longer or two. But the uncertainty in their age of death is not going to change. And so I think if you added that kind of thing to your analysis, it'd be part of the additional information. The other thing is, I think, to Amal's point, we do studies on samples which are sort of useful and from which we then generalize dramatically, like your sample. I mean, I've been to Dunedin. I have a good friend on the faculty there, and it's a small, cold corner of a small, cold part of New Zealand. It's beautiful. It's gorgeous. And you can see very interesting seabirds and that kind of thing.
No, they all immigrated from Scotland within a 50-year span. So
you can say much the same about Scotland. The point is, I think it's a great sample because of the nature of the study. You know, so it's phenomenal from that point of view. But in terms of representativeness or representation, I don't know. It's the best we know. So I'm going to cite your study and say, you know, this is what we think. But if I was doing a study, if I was trying to apply those lessons to Atlanta or something, I don't know. I assume it's going to work.
We're going to find out.
Yeah, I mean, I think this is actually a really interesting area. I mean, there's no reason that the process through which one obtains education and then translates that to other success has to be the same in New Zealand and the U.S. I mean, and those are two relatively comparable countries, so you can imagine.
Yeah, I mean, I think that's an interesting question. You might get at with some VED help.
Still planning to.
We're working on it.
It's in the works.
Yeah.
Well, I have a different kind of question, which is much known about how to use something like someone's Safeway purchase barcode data or the Amazon data in relation to some question about, that would be useful in the context of the studies we heard this morning.
Right. So let me give you an example. So this was a study that we couldn't do, but tried to do. So Safeway asked us to study a very innovative and quite aggressive wellness program that they did among their employees, and it took 4 years before they showed any benefit. It was a results-driven, highly economically incentivized program to get your weight down to normal, to get your blood pressure down to normal, to not smoke. You got rewarded. It didn't matter where you started. There were benchmarks, and every year you got examined. So they collected all fabulous biometric data, and they were able to show they cost them money at the beginning because people got into treatment for various things that hadn't, which was probably good. And by 4 years, they actually were starting to save a little bit of money. What was really would have been fascinating, but they wouldn't give it to us, is the consumption data. And the reason the consumption data would have been very valuable is because they get a 20% discount. The employees get a huge discount. So it seemed extremely plausible that they buy almost all their food there. So that we actually could, because we had enough information on how many members there were in the family, we actually could use, and when you go to the market, you use one of those little swipes, right, for your whatever. So they basically had a complete list of everything they purchased. And we could actually then have answered a question like, did they do better for medical reasons? We had all their drugs and their medical visits, or did they do better because they actually changed their behavior, ate differently, and things like that? It answered an important question about a wellness intervention that otherwise would have been unanswerable. I don't know anyone that's actually pulled it off, because you couldn't just do it generally in the population for many things because people have too many different ways of achieving the same results. If you wanted to study a society's book purchasing, I mean, Amazon would be a great place to start, but it would be far from a complete registry of every book that changes hands. And so that's the problem. Someone's going to have to be clever about using those data in an appropriate environment where they might indicate something about behavior that you otherwise couldn't observe. I mean, so there's no magic. The burden falls to us as investigators to think of cute ways of linking.
So it seems to me one would probably want to start by constructing a dozen indicators based on this mass of data. And then what dozen indicators? Well, probably they would be inspired by kinds of questions that are asked in surveys already or something like that. I don't know. And I imagine it would be some years before anyone would figure out how to exploit the actual new richness in these data. And it's a little bit like the GWAS or something, I
At
suppose.
the beginning, yes.
And
which is you do it unsupervised and you see what the data look like and you say, what's it telling me? And then you've formed some hypotheses. Yeah, I agree.
So I'm just wondering if anyone's doing that sort of thing now, or if this is all just sort of, it might be nice in the future, then we can figure out how to use it.
So who do you mean by anyone? I mean, so remember, our friends out there are not only collecting that vast amount, people at Google and Microsoft and all the people who run, web browsers and things like that are collecting phenomenal amounts of data and doing a phenomenal number of experiments every day. They're doing thousands and thousands and thousands of mini experiments. Well, so they're not interested in health yet.
Yeah.
But the day may come when
But the principles would be the same, because projecting the next kind of book that someone might want to buy seems to me quite different.
Oh, I totally agree. The predictive, I mean, so yeah, I don't know of anyone that's really actually made a study of what from that form of administrative data we could borrow and really effectively use in trying to unravel the life course from the point of view of the questions that matter most to us. But it's for lack of trying, I think, not for lack of there being potentially available opportunities.
Another thing is like health and profit are working orthogonally, right? Because cookies have high profit in them. So, but I mean, and for instance, like in Target, I heard a talk from the president of Target. Apparently, pregnant women are a gold mine. That's the time when most people change their habits, and you want them. That's the time. So you make them buy their stuff and then cross the cookies aisle and the juice aisle and everything like that. So ensure they pick up everything on the way because they're tired. And so finally, I'm going to pick up this and that. That's how you develop purchasing habits.
They know a ton about health. They have the bathroom there.
They have it all.
And they certainly can predict pregnancy way before your doctor, because they know very early on the searches you're doing and they're very good at that.
I did want to add one more thing to the selection issue, because that seems to me something that's come up that many of us around the table were not working very hard on before, and the juxtaposition of the conferences we actually had here in this room and at the site opens a perspective. The value of randomization in the classical experimental designs is not controlling for the things you know about. It's controlling for the things you don't know about. And so while I strongly agree with Ben that representation rather than representativeness is key for many purposes, it does not get at the
unobserved stuff.
Unknown, poorly understood reasons that people are in the UK Biobank. All those voluntary decisions that lead them to volunteer. However, the folks at the other meetings are worrying a lot about that, about these more complex selection issues, and machine learning opens up at least a spectrum of things to try out. Not all of it working, but with split samples, you can sometimes see whether machine learning bases for letting the data guide you into what seem to be determinants of selectivity. So you get not the controls on the variables we know, but a set of complex controls on constructed variables that potentially can be used for reweighting and stratification. So there is this world out there, and a lot of these people are at Microsoft, Facebook, and the big centers we're talking about. Seems to me the world of this conference and the world of the last one would really benefit from coming together.
Sounds like a great next conference. Maybe that's a good segue to lunch. What do you think?
Yeah.